All right, I think we can go ahead and get started. So thank you all for attending this talk. I'm glad everybody was making it with the reschedule. But yeah, so today we have Angus Ingermeyer with us from the California Department of Public Health. He's going to be talking about bioinformatic pipelines used to determine close related non-tuberculosis mycobacteria species. So you can go ahead and start.
Hi, everyone. Can you hear me? Great. Yeah, thank you so much for coming, everyone. I'm Angus Engermeyer. I work at the California Department of Public Health in the sort of new-ish Genomics Center, which is a group that we put together to try to provide sort of a core facility in-house for sequencing capabilities as well as bioinformatics analysis. And a lot of what we do is help other groups with some of their bioinformatics programs, since as as bioinformatics has increased in importance in public health, a lot of groups have kind of had to find their own way. So our approach is to try to at least not take away anyone's um, work or responsibilities or anything, but try to provide some support where needed. And that can be some pretty small projects at times and bigger ones like this one, which I'm going to talk about today. So in this project, I was approached by Vivara Kotureva and Swarthi Rao, who are in the tuberculosis group. And one of the problems that they face is that a lot of infections that look like tuberculosis or might kind of seem like they're related, either by sequencing or some of the older amplicon-based methods, um, but aren't really panning out as tuberculosis. And they fall into this group of non-tuberculosis mycobacteria. And it's a diverse group. It's a varied group and it's got some eccentricities. And so we wanted to work on a pipeline that would kind of help parse some of that out and be really specific to NTM. So this is definitely a work in progress, and it's a little bit in the mid stages of, well, it's the late stages of development, but it's definitely pre-alpha at this moment. The keys work. So yeah, so I'm going to talk a little bit more about why this NTM pipeline is needed in the first place, some of the specific bioinformatic difficulties inherent to NTMs. I'll talk a little bit about the pipeline. I'm not going to go into a huge amount of detail, but can I keep a more of a conceptual overview, and I can answer any questions about it. And then a couple of aspects of the pipeline that are really specific to NTMs, why this hasn't been totally just an off-the-shelf pipeline, is the species calling model and some of the validation that we've had to do with that. I'm sorry, it's popped up in my face. Okay. So an overview of NTMs is that they are of significant clinical relevance. And this is something that's been known for a few decades, but it's really been increasing as more has been studied. And I'll hasten to add at this point, I'm not an expert in this. I have a PhD in sort of like medical microbiology, ecology of microbes. And so I know a little bit about a lot of different things, but I'm definitely not a TB or a mycobacterium expert. So a lot of this is kind of regurgitating what I've been told by my colleagues and kind of some of my, surface level analysis. But for the clinical relevance, these can include things like TB, pulmonary disease, soft tissue infections, some central nervous system things, bacteremia, ocular diseases, and a range of other things. A lot of these are severe pathogenic diseases, and some of them seem like they might be a little bit more opportunistic. But that's kind of the themes with NTNs, is there's a wide range of things they can cause and sort of their genomics in general. This is a particular concern, and I think Rivara's main point here is that she's seeing a lot more of these kind of infections come through. So they work closely with clinics that will get patients with these TB-like diseases or mycobacterium-like diseases that aren't totally identifiable. And along with the global increase in rates, so mortality is up over 2 times in the last decade or so. In California, I think they're seeing much more clinical infections as well. So we need to find this way of quickly analyzing, sequencing the cultures that they get from the clinics, analyzing what they could be, and try to get that information back to the clinician in, soon enough so that they can actually put in place some of the treatments. And that's really important because what is known about some of these different species is that the treatments can vary. Right now on NCBI RefSeq, there's about 250 of these curated NTM genomes. That may include a couple of duplicates here and there and a couple of things that are really more tuberculosis complex related. There's a little bit of gray area there. And I will point out that there's a couple of subgroups that are much better known that cause the majority of the disease, but there are lesser ones known too. So if we look at the overall phylogeny, on the left here, you can see some of the members that I've rooted this with of the mycobacteria and tuberculosis complex. That includes tuberculosis. This one has Candidii. And there's a number of those, obviously, and I'm only showing just a couple as an outgroup for this NTM table. And they're fairly distant from the nearest NTM in this phylogeny, but the NTMs themselves are widely diverse here. So identifying the species is, kind of the point of this whole thing. One of the big problems, though, is that these two groups, so the Mycobacterium avum complex is the #1 driver for infections, and then this Mycobacterium abscessus complex is the #2 driver. And you can kind of see, and now these, don't read too much under the phylogeny, just kind of give an overview. But you can kind of see that this MAC complex here is much less diverse overall than just kind of the average NTMs in general. And that's even more true with Mycobacterium abscessus. There's 3 main species, and they're shown here: abscessus, or subspecies, excuse me, abscessus, Bolletii, and Mycolense. And those are very, very related. And so one of the issues with this pipeline is that it's been difficult to not only analyze a number of species that are highly diverse across the range of NTMs, but to also correctly identify specific species within these much more closely related groups where the treatment, the clinical treatments may actually differ. So it is pretty important to get those answers. One caveat here is this is actually based on just one of the target genes I'll talk about a little bit more later, but broadly, the genomic phylogenies are very similar to this. So like I said, TB is distinct from NTMs, and it's about 83.5% average nucleotide identity to the nearest NTMs. And there's about an 80% ANI identity among all of them. So it's just, so the NTMs themselves are quite diverse. And like I was saying, within the complex, especially the M. abscessus complex, there's actually 96% similarity across those 3 at the genomic ANI level. And that's above a 95% threshold. I'll talk about a little bit more too. That's commonly used to define species. So it just kind of gives you an idea of the scope of the problem of what I'm dealing with for this pipeline. Some of the existing sequence data we have is strongly focused on the abscessus, or the avum complex, and the abscessus complex, because like I said, those are the 2 leading drivers of infection. But a lot of the data on the SRA is, they know like this may be an infection caused by this, but there's a lot of co-infections in there. Sometimes it co-infects with TB, it often co-infects with other NTMs. And so pulling those out is really important, especially if you're looking at like a different type of antibiotic resistance profile between the 2 things co-infecting. You need to know what both might be in there. And validation is kind of a rough term. It doesn't necessarily mean much, but it's just to say that a lot of the sequences out there are kind of data dumps, which is really helpful. But for developing a pipeline where I really want to know if my outputs are correct based on my inputs, I don't have a great idea of how correct all of the inputs are for existing sequence data. And then one of the other bioinformatics aspects of this is that historically, a lot of these NTM identifications, well, all the identifications have been based on this 2 target genes, a heat shock protein, which is just a chaperone, highly conserved chaperone protein, and RPOB, which is an RNA polymerase, which is also pretty highly conserved, maybe not quite as much so. But those 2 have been found to not really have enough discriminatory power to identify between all the NTMs, especially within these the various more related complexes. My colleague Spurthi Rao, she's an expert in abscessus, and she has been doing this work on including another gene marker that's tlyC, which I can't actually remember exactly what it is. I'm not sure if it's been super well characterized. I know it's a hemolysin in like rickettsia, but it does have like some cell wall associations, I think. Anyway, uh, it is a slightly more diverse biomarker. It's conserved in the sense that it seems to be present in all of the NTM species, but does diversify, biologically more rapidly than some of the more conserved, like HSP65 and the polymerase genes. So just to give like kind of a really high level overview of where the pipeline is at right now, I'll just kind of briefly walk through it. And I should also say, if anyone has a question, please interrupt me. So I'm trying to make it work with a number of different sequencing platforms. So Nanopore, Oxford Nanopore, as well as Illumina, and each of the various chemistries and versions hopefully should be accepted. So that's run through a quality control and read filtering step. And that is going into a sequence assembly step. So it's all kind of a very basic thing at that point. One place where I branch out here is calculating the actual coverage and the masking, which I'm going to talk about next slide. And that's a way of editing the actual overall assembly to reduce like positions where there may be low coverage. So that's the main step here to develop a whole genome. The whole genome sequencing side is on the left. And then the right side is we actually, in addition to the whole genome sequencing, we also want to keep using these targets, which gives us a little bit more discriminatory power. It allows us to do these kind of species typing analyses in situations where the sequencing may be of a low enough quality that the whole genome assembly isn't really good. And it allows us to compare the data we generate with this kind of pipeline with historical data that may be based just on the target amplicons. A couple of the aspects of this is, like I said, I want to keep it sequence platform agnostic so we can bring in as much data as possible. And also, like I said before, that different groups are kind of doing some of their sequencing. In our group, we pretty much do only Illumina. Some other groups for cost saving measures have their own ONT setups. And so we end up getting like a ton of different data from different platforms, different chemistries, and different qualities. And so I'm trying to be as agnostic as possible to like accept all of that and do the best we can. Excuse me. Currently, it's based on the RefSeq database, like I was saying before, of NTMs, and I just wrote this so that could be updated. When I started this project, there were like 217 NTMs in the database. Now they're up to about 240. So that's something that is going to need to be updated pretty soon. The typing targets are focused on those three, but the way the pipeline is written is you can add in any number of genes you want, and it will try to use those as typing targets as well. So once you have your data assembled into your whole genome, and then you have extracted your targets from that, those both separately go into average nucleotide identity calculations, where it looks at the DNA sequence of those compared against everything in the NTM databases. And TB is included in there, of course, because you want to know if you have a TB co-infection. It computes those as best as it can, and then that gets fed into the model where I try to make a final decision about what is going on in the sample that I've been given. And of course, it's nice to go into a LIMS-friendly output so laboratorians can take a look, confirm it, and put it into their database as necessary. The calculate coverage and mass step I wanted to point out because it's kind of an interesting thing. Everyone has their own cutoffs and thresholds where they like to validate. And historically, a lot of the TB pipelines, at least at CDPH, have a hard cutoff of 10x coverage. And some of them are very strict, so that if you see any nucleotides below 10x coverage, you actually throw out the data essentially. And what I found is looking at a lot of these samples based on, and we haven't totally figured out exactly what this happens, but it happens frequently enough to be a problem. We'll get exceptional coverage. So this is averaging, about 100 or 120 or so across the gene. And this is just one of the target genes. It doesn't really matter. It happens to different genes at similar rates. But you'll get these tiny little dropouts just for sometimes just a couple of nucleotides or a single nucleotide that will dip just below this cutoff. And so early versions of the pipeline where we implemented existing thresholds and cutoffs that the group likes to use was throwing out a lot of data. And so we're kind of testing this approach where at the sequence assembly step, well, just after the sequence assembly step, I remap reads back to the assembly and look for these low coverage areas. And any nucleotides that drop below the threshold, in this case 10x, we actually just mask into ends and keep the data otherwise. And this works out pretty well because if we're looking at, if the final step here is average nucleotide identity and you have too many ends, your average nucleotide identity is going to fail at the thresholding step for ANI. So you don't actually need to throw out this data at earlier points. This is obviously an extreme example where, like, you can very clearly see that 3 base pairs here that dip below the 10x coverage aren't going to affect the overall analysis. But there are times when it's, what happens when you get into, 1% of the things, 5%, 10% of the base pairs drop below your coverage levels, then you're obviously going to have to decide at what point it gets cut off. And that's what the ANI thresholds will do. So the next step is to take a look at the model. And so this is a super high level concept of how the model works. It's pretty much just a series of, if-thens within the code. But to conceptualize what it's doing is, essentially we take the FASTA files, look at the quality of those, we generate an assembly, look at the quality of that, and then we do the whole genome sequencing ANI. And so if you get a whole genome sequence out of this, and it's great quality, and it beats this, wall of 95% threshold, which is what we've arbitrarily chosen as the whole genome sequence ANI. Then we can just call species ID. You don't really need to muck about with the targets or anything like that, because whole genome sequencing of high quality is pretty much the gold standard as far as we can do right now. There's a few where this can be, stopped though. If we get poor read lengths, or low read count, low Q scores, that can stop this analysis. Although in reality, that pretty much just stops the assembly step. So it's kind of, drawn a little bit wrong here. And similarly with the assembly, if we get assembly that's too short, indicating we just don't have enough length coverage of the genome that we're trying to assemble, we're not going to get a good enough whole genome sequence ANI. And that's really important because the way ANIs often work is they don't really take into account the relative lengths of, your query genome and your subject genome. It just looks at what the similarity is of the overlapping areas. So it's really important to make sure that, you're comparing within 10% or so, usually what I say, similar lengths of your two genomes. So if you have a reference genome that's 6 megabases or whatever, but your whole genome sequence assembly is only 3 megabases, even if you have a very high ANI, 99% or whatever, we're probably going to throw that out because that's just not enough whole genome sequence data, obviously. And similarly, too long can be a problem. If you're getting co-infections, this one gets a little bit trickier, but if you get co-infections, sometimes it can't distinguish the contigs from one another when the potential co-infections are very similar. And this is mostly a problem with Mycobacterium abscessus group. But there's a little bit of extra logic that goes into parsing those out. And then obviously low N50 is just a rough metric of assembly quality. I also wanted to mention a little bit about the 95% threshold. This is something that a lot of people use, and I know thresholding can be a controversial topic about exactly where people set them. There's a recent paper I like down here where they compared about 20,000 existing genomes in GenBank and found that at 95%, it pretty much captures intraspecific similarity really well among almost all the species that they look at. And then below about 90% is where it's a really strict cutoff for different species. So at the moment, I'm using 95%. That's obviously that's tunable. And I'll talk a little bit more later about the validation. But that's kind of what I'm starting with for the threshold on this. Oops. And so, like I said, yeah, the 95% threshold is a problem here for no problem distinguishing TB from NTMs when they're 83.5% ANI, no problem among most NTMs, but then when you get to the complexes that are about 96% similar, obviously you can't separate those. They're different subspecies, species, it doesn't really matter. They need to be identified anyway. So then that gets into what to do if we failed some of these steps. So specifically, if you fail the 95% threshold steps, we still can go to the various targets. And this lets us look at the same exact sort of thing. So this also applies to like maybe if you have too short of an assembly, even if you ended up with like 3 megabases and you expected 6 megabases or something, you might still have sequenced and been able to recapitulate in the assembly the 2 or 3 of these genes, in which case we think that's enough to move ahead with the species identification. Each one of these has to pass a similar thresholding check. And determining those exact values is a whole other thing that I will talk about as well. And the larger circuit here is not that it looks fancier than it is. It just means that we're starting off with you have to have 2 of these beat the thresholds, and that's enough to consider a species ID, not all 3, and not 1, obviously. So I wanted to talk a little bit more about these 3 targets and the diversity among them, and just how that is implemented bioinformatically here. So what I'm showing here is a clustyle diagram zoomed way out of each of these genes. So what you're seeing is that each one of the rows across is one of the 217 ref-speak genomes, and these are all aligned to each other. And then the columns are each base pair positions. You can see the scale at the bottom here. So HSP65 is about 400 base pairs. Well, the partial gene that is looked at here is about 400 base pairs long. And if I remove that. So what you can see here, and like this is definitely like, 50,000 foot view, but you can see that there are broad strips where there's a lot of conservation. There's regions of higher conservation, there are fuzzier areas where there's lower conservation within the HSP65 gene. And that's also similar for RPOB. There are these broad regions of conservation. There's a big gap insertion area here, which I think is why I'm getting some of the weird coverage dropouts of that area as well. But some of this conservation, like I was saying, has led to, is the reason why HSP65 and RPOB don't have the discriminatory power always to distinguish between closely related NTMs. So the third target gene here that Swarthy has implemented, you can see just right off the bat, it's a lot fuzzier here, it's a lot more diverse, but it is conserved in the sense that it is in all of the NTMs. So that one adds a lot of extra power in trying to distinguish species from each other. And coming back briefly to some of the work that I've done on trying to analyze the thresholds for these, there's a lot of work that's been done on determining whole genome species thresholds, like this 95% threshold, and there's other ones, other papers out there. But for individual genes, it's a messy area of the literature, and there's a lot of argument about where those cutoffs should be. A lot of people say about 80% is good for a gene, though that varies wildly when you're looking at genes of different diversities. That may apply pretty well to things like 16S or HSP65 or even RPOB, but for tlyC, it definitely does not. And so we wanted to take a slightly more sophisticated approach and try to tune those thresholds to meet our data specifically. It's by no means applicable to like a broader range of organisms or other pipelines, but within the NTMs, we're hoping we can target values that are meaningful. And one of the ways that I've been approaching this is to look at a comparison of the average nucleotide identity similarity between the genes as compared to the whole genome AENIs. So all of this shown here is for all the 217 ref-seq genomes, we did a pairwise comparison of how similar the whole genome is on the x-axis, and then for HSP65, how similar the HSP65 gene is, and then those all get plotted. And you can see as you're in very lower levels, 80% of whole genome ANI, it's just a big diffuse cloud of possible HSP65s, and that's the same for RPOB and tlyC and essentially any gene. But as you get in higher rates of discriminatory power, where you're getting to that 95% threshold, you're seeing it kind of linearize and become something that we can look at a little bit more closely. So if we zoom in on this region, and this is some early work where I was looking at about 85% to 100% whole genome ANI cutoffs. So I'm zooming into just this region right here. So I plotted a linear regression along the data in this area from 85 to 100% whole genome ANI similarities, and was like, naively, I was thinking, well, maybe we can look at what the threshold it could be at the intercept there. So like the average threshold intercept at 95% whole genome ANI, which is again where I'm making the species cutoff determination, we're seeing a intercept for HSP65 specifically closer to 98.4. And I was like, okay, well, that's interesting. And that's maybe what you'd expect. Well, maybe it's not exactly what you'd expect. I don't know. This was a little bit of a lark. But it did show me that there is this area, right, 95%, where the average HSP65 ANI is a little bit more stringent. Unfortunately, what that does is everything to the right of this vertical blue dash line here are valid species comparisons. So these should all be, all these points are pairwise comparisons that should be considered the same species, essentially. And so you can see here, if I put the cutoff at the blue line, the horizontal blue line, which is the intercept at 98.4, there's a few comparisons below that where they would be excluded at HSP65, as they would be considered different species by HSP65, but the same species by whole genome ANI. So this is kind of on the back burner, except for the fact that I've kind of just using these pairwise, the lowest pairwise comparisons that I see as setting the threshold. So the threshold for HSP65, I'm usually setting at about 94% to capture these lowest two. So it ended up being a little bit less sophisticated of an approach than I had hoped. And it's going to vary depending on the pairwise comparisons of the database. So this is all based on my earlier RefSeq database of 217. But now that it's closer to 240, I'm going to have to redo this and recalculate what those thresholds would probably be. And just to show, these are all of the three genes, but it's a very similar concept. tlyC, actually, the intercept threshold is very close to the lowest seen value. But for the other twos, there's some stragglers down there. So just to revisit the model and a number of the variables that are going into my consideration of this is, like I said, read length, read count, Q-score, all of these FASTQ quality metrics, the assembly metrics, the whole genome identity, the whole genome coverage, which in this case, I mean, like essentially how much comparative length there is for the ANI. So when I was talking about a 6 megabase genome versus a 3 megabase genome, that would be ruled out, but a 5.9 megabase genome, if I see that in my data, and then the reference is 6 megabases, that is going to be accepted. So there's a threshold there I have to calculate. And then for each of the target, there's a percent ID at these walls here, X, Y, and Z that I need to calculate. And then the similar aspect for the coverage, make sure I have enough of those genes. And so that actually ends up being quite a few variables to consider. Excuse me. So I need, some of these I can make pretty good guesses at. Some of these assumptions are pretty basic, but some of them can widely vary. And with every additional variable that I need to create a threshold for, validating this pipeline can get a little bit more complex, almost exponentially. And to revisit some of the problems with existing sequence data is, like I said, it's really focused heavily on a couple of those complexes, on the whole NTM set in general. It can have a lot of unlisted co-culture and co-infection kind of problems. And again, it hasn't been validated like a public health sense in most cases. So what I wanted to do is develop a kind of a regulated validation training set. And this is kind of a whole other rabbit hole that I've gone down on this project, um, largely because of those previous issues where there's not a lot of existing data. That's great. Um, I'm trying to build essentially in silico data that pretty closely recapitulates what we're likely to see based on some of the clinical data that we have, which we also don't have a ton of, but it's a starting point to try to figure out what these variables should be. So, like I said before, we want to look at Illumina and ONT in the future. Also, hybrid of these. Sequence quality, coverage, and depth are both things that can vary here. The number of strains that are potentially within the data set are going to be varying. And then other considerations like, whether or not there's a secondary NTM co-infection within within like a validation sample. And I should say these are all in silico samples that I'm trying to generate right now. So you have to consider whether or not there's a co-infection strain, what that co-infection strain would be, like how closely related to the primary strain that I'm testing, and then the relative abundance. And you can see right there, that's getting to be quite a number of variables. And the same with TB. So it's really important if we run the sample through, we have well-established TB pipelines that can do a lot of the analysis for exact lineages and antibiotic susceptibilities, and that's really important to get back to the clinicians. But in the situation where we might think it's an NTM, we can run it through, we want to be able to make sure that we're also detecting it's TB, so that can get shunted over to a more advanced tuberculosis pipeline. So in these validation training sets, I also want to include potential TB co-infection, and there's like 7 kind of lineages of TB that might be considered here. I'm starting with just one. And then, of course, the relative abundance of tuberculosis. If it's at 0.1% of the sample, like, am I realistically going to be detected? Maybe, and it's pretty important. So maybe I should be planning for that. But if it's 10 to 50% of the sample, then obviously we definitely need to be detecting that within the data set. So just kind of give a range or an idea of the range of variables that I'm working with here and trying to build this data set, it gets pretty significant fast. So, Illumina versus nanopore, C quality, that's, good versus okay. And the details of that can highly vary, of course. Like I picked, for okay, I picked 1.5% of genome below 10x coverage and an average Q-score of 15. And then good is higher coverage and more adequate Q-score, but those could be wildly variable too. One of the big restrictions here and why this has become something of a difficulty for me is that there's 2 or 3 main existing pieces of software for generating in silico reads, and they all are okay. Some of them are pretty good. But they don't have a lot of nuance or sophistication. You can't tune like the sequence quality very well. Like kind of the best one doesn't let you doesn't generate any kind of variable length within the sequence read. So you can say, here's the quality I want, here's the co-infection variable abundances that I want, but it'll make every single read exactly 250 base pair paired or whatever. So one of the things I do want to do is kind of use some of this modeling that I'm doing and some of the existing clinical sequence data to edit those in silico reads to make them look a little bit more realistic. But for the moment, this is, a little bit canned. So in addition to the sequencing quality, right now I'm just looking at one primary strain and abscessus, subspecies abscessus, which is kind of why this whole project got started. So I started with that, but obviously you want a validation set to choose, I'm probably going to want to choose 10, 20-ish NTMs from a diverse segment of the overall phylogeny of NTMs, especially including the Mycobacterium avium complex. And then if I include a second strain, which at the moment I've just chosen one specific strain, M. chelonia, which is the most closely related to M. abscessus that's outside of the complex. And then you want to vary the relative abundances of that strain and then consider if we're doing a TB co-infection, varying the relative of variance of the relative abundance of that. And then finally, cases where we want to have, essentially a tertiary infection where it could be M. abscessus, M. cheloniae, and then a tiny bit of tuberculosis. I'm not sure how often that happens, but I know it can happen. So we're trying to consider it. So that's just sort of the initial data set that I was trying to generate. And I've run that through the pipeline and it generates some answers. So kind of what you're looking at here is a tiny subset of all of the results data where I'm looking at some of the Illumina reads. These are the higher quality ones with an average T-score of 32, 500,000 reads versus a million reads. And then the varying abundances of M. abscessus, M. cheloniae, and tuberculosis in here. And the results are, whole genomes. You can usually recapitulate what is going on. There are cases where it's problematic. There are obviously cases where, especially, you can see here that the M. abscessus subgroup, so that's the 3 different subspecies of abscessus, all show well above that 95% ANI threshold. So that requires a second step when it's abscessus to start parsing those out. When we had a 50% mix of abscessus and cheloniae, similar, we're kind of recapitulating with both, but you still have to do that abscessus step. And then as the sequence quality gets worse and the mixes get more complicated, things started to fall apart. This is a little bit older, and we fixed some of these. Some of it's bugs, and some of it is just, threshold problems and bioinformatics issues that need to be parsed out. And so I think we're getting cases where we're mixing reads from tuberculosis and some of the other NTMs and coming up with totally incorrect identifications. So that's a problem, and that's something that needs to be addressed. So the main point here is that obviously work to be done. And I really want to bias this model and the thresholding in a way where we can err on the side when mistakes are made of false positives, which are still not great, but try to minimize those as much as possible, but really, really try to avoid false negatives, since these are clinical infections and they're often quite serious, and the clinicians need to know what's going on as soon as possible. And so hopefully we won't want to miss something that's medically important. And with that, I just want to thank a special thanks to Varvara Kozyreva, who really is guiding me on the mycobacteria aspects of this project and the details of validation that they need for their group. Smriti Rao, who's an expert in NTMs and especially the subspecies subcomplex, and John Bell, who I work closely with in bioinformatics at CDPH, and then the mycobacterium group and my CDPH Genomic Center. Are there any questions?
This is Dwani Bhatt here. I actually have a question about the 10x. Especially if you can pull up the coverage for the RPO that you showed, that might help with certain things in context there.
Sorry, is it still sharing? Is that correct?
No.
Oh.
And as you're doing this, I know thank you. This is lots of good work and a lot of due diligence that has gone in there. So thank you for walking us through it so well and all the work that's gone in there. The question I have is around 10x, is when you see these 1Z, 2Z base drop off, how do you know that it's not real deletion? I don't think the 10x coverage cutoffs were meant for a single base, because then you'll miss deletions, right? Like, how would you treat them?
Yeah.
I just wanted to, and I know there's a lot of people on this call, so others might have some thoughts about it. I think 10x is designed more for a broader drop off, like we couldn't even get any sequence across certain regions, not for these onesies, twosies. So I was very curious, like, what was a step that was filtering out a lot of data just because there was a drop off of 2 base or a single base pair or a very short stretch, which can be a real deletion, actually.
Yeah, no, that's a really good point, and there is definitely some work to figure out there. I will say that if I see 1x or 2x, I think the threshold is 2x coverage. I will throw out that data because we just don't know. Um, these are cases where it's like dipping into 9, 8x, and we really aren't sure. But it is based on the sequence assembly. So in theory, we're probably getting the correct data. The big caveat, of course, being that if there is a co-infection that are highly, highly similar, and
Hmm.
there is, one of them has that SNP that you might be talking about, and the other doesn't,
Hmm.
then yeah, you could be getting a mixed assembly, and you could see something like this. That's something I have ruled out for a number of the abscessus strains that I've parsed through.
Hmm.
But it's yeah, it definitely needs to be looked at. This doesn't happen very, very often. I'm only seeing this in like 5-10% of some of the samples of lower sequencing quality. Usually it's just not an issue. Um, but it is a good point. And there's 1,000 little things like that I kind of have to make some assumptions about. So for the moment,
Right,
that's
right.
kind of where
And
it's
I would
at.
like to hear from others too, like what they think about the interpretation of this 10x cutoff. I mean, if you're doing a reference-based calling or something like that, then you would, it depends on your method, because if you were doing de novo, you would probably take all your reads, you'll let the assembler make all those calls and kind of work through it. But if you were just having these onesie twosie and you're doing against reference, then you often usually say, hey, to make a base call, you need at least this much read coverage, and then you go for majority or something like that. So if you say 10 or whatever the case may be, I mean, um, uh, otherwise you would say an N or an indel there. So I'm wondering like how others are thinking about the 10x cutoff as a whole and the application in this case, because I think as a group we need to understand this 10x much better than we do. I guess nobody else has that question.
No, it is a good
Um,
question. I think one thing that would help is to look at a number of other examples and kind of put them up on
right,
a presentation
right,
sometime and try to get an idea of what the biological reality of these might be.
right. Saying what cutoffs we're using and what they actually mean. Because in this case,
Mhm.
it's almost like there's some kind of mixed population in here or something, some with deletion, some with not. Or is it all
Mhm.
sequencing errors? Plus, it's also interesting how the coverage kind of falls as you go across the whole gene. It's just
Yeah.
maybe it's just one of those kind of depends
Well,
on
you can
what
see,
methodology was used.
yeah, for sure. And you can see this weird dip here that like
Hmm.
is also similarly problematic looking, but does stay above the coverage. So, another problem with
Correct.
thresholds is we're just ignoring
Right.
that and saying that's great, which is probably not true. And if I go to look, so this is RPOB. If I look at
Hmm.
RPOB right around that 400 base pair position, there is this area
Hmm.
where there's a few crazy insertions in a number of species. Well, a small
Hmm.
number of species. And so there's probably something going on around there, whether it's indel related or
Hmm.
there could be some sort of insertion, replicated insertion in cell element. Who knows? There could be some biological explanations for things like that, but also things like
Right.
this.
Right.
One base
Right. It's the
pair
same.
makes me
It's
a
just a
little
scale of
suspicious, but
coverage
yeah,
across. Yeah. Hmm.
yeah.
And then just one more confirmation where
Mhm.
you have that two cutoffs in your graphs, the linear and your 95, 98 across HSP. This
Yeah.
is just for me for confirmation in my head. Is this, am I thinking of it accurately? The only false negatives would be the one in the bottom left quadrant of your two blue line quadrant, right? Because if you were using the 95 ANI cutoff, everything to the left of that vertical line would
Hmm.
not be. Everything to the right of that vertical line would be considered same species. The ANI is high enough,
Yes.
except your cutoff of 98 would rule them out. So the only thing, the false negatives are in that bottom left quadrant.
Yeah. And so, like I said, actually, I'd drop this blue horizontal line down to capture these two small ones down here.
Okay.
So we're getting everything. So yeah, so there is some confusion. Like, yeah, there are
So
areas where there would be problems like that. But
okay.
that
All
would
right.
be to reduce false negatives. I have to drop that down.
Mhm. Okay. Thanks. Anyway,
Mhm.
those others listening, if you have some thoughts about 10x, I would love to have that discussion, but some of the time maybe. But think about it. It's just
Mhm.
interesting to see how to interpret these things. And that 10x cutoff has become lingua franca, and people that automatically use
Mhm.
this to, and what are we gaining by that? And is there saying give?
Any other questions?
So I have a question. I'm Emily from UVA. I'm going to fully acknowledge that I missed the first 10 minutes of your presentation because I had to walk over from another talk. So please correct me if I'm way off base. But I was wondering if you could just comment kind of on the extent of the reference databases that you're using for RPOB 16S and then tlyC. So I hadn't previously heard of tlyC. Do you have a lot of genomes or a lot of other tlyC genes that you can compare to? And can you just speak to the diversity of those databases?
Yeah, so the database is built, if we go back here, the database is all of the NTMs that are deduplicated on RefSeq right now, excluding, well, yeah, all the NTMs. So I have to search for whatever the tax ID is for mycobacterium and then remove the mycobacterium tuberculosis complex and put those in different databases. So anyway, these are all essentially all the NTMs that are curated and have high quality genomes. Um, every one of them possesses the three genes that we're talking about, which is the heat shock protein, the RNA polymerase, and this tlyC. And there's probably a whole bunch, a ton of other core genes that are in there. And yeah, what I was actually saying is that my colleague Swarthy has added tlyC as one of her big projects, is to use tlyC essentially as this third gene to help with the diagnostic differentiation of the various NTM species. So historically, it's been HSP65, and RPOB were used exclusively in an amplicon-based format to determine which NTM species you're looking at. But that was found, especially in these more similar complexes and similar areas, to not be enough discriminatory power. So tlyC adds a little bit more. It's a little bit, there's always kind of a trade-off when you're looking at the genes, because the more conserved the gene is across all the species, like the higher quality it's going to be, but the more diverse it is, the more discriminatory power it's going to be. So tlyC seems to be in an okay spot for that. Like I say, the main thrust of this pipeline is, I think 99% of the time I'm going to be getting enough sequence data of a high enough quality just to do a whole genome sequence analysis. ANI, call it very simple, done. But using these three marker genes allows us to have like a little bit of backwards compatibility with historical records, and a little bit more discriminatory power in cases where there's some confusion.
And then are you guys reporting this back out clinically, back to laboratories? Like, would they be able to send a clinical isolate to you? And then in a patient, let's say that has COPD, send another clinical isolate to you for identification later on to confirm it's the same thing prior to starting treatment?
Yeah, my understanding is that's the idea behind this. I think they're seeing a lot more NTMs on the clinical side in California, and this has become more of a concern. So they want to have a pipeline set up that's like on Terra or wherever they want to deploy it. So it can pretty rapidly just jump the sequencing in there, get an answer, send it back to the clinician. The big caveat is I'm not on that side of things at all. I have no access to any kind of clinical information or even really how that all works. That's within the NMDPS division that Vivara is in charge of.
Thank you. I just find it, I mean, the RefSeq database is so limited, right? You only have 217, and yet there are thousands of non-tuberculosis mycobacteria. So when you have these patients that have cystic fibrosis or COPD, what we see in the clinical labs coming back out are NTM species that like we hadn't really ever heard of or seen previously. That might
Well,
not
that's
be
really,
well
yeah.
represented in this database. Yeah.
Yeah, that's a very interesting point. So, I mean, obviously I can put in any database I want into this. I wanted to start with RefSeq just because, I can be assured of the data. I've had a lot of problems in my career, like using larger, less curated databases, and ended up finding out that a lot of the metadata is wrong. or there's a lot of duplicated problems, or it'll end up being 30% human DNA or whatever, and just got dumped into the SRA. So I am a little bit cautious about that, but this is mostly for development, and that's a really good point. In fact, I'm going to write that down and just try this with larger databases too to make sure I'm capturing enough of the diversity.
Or sometimes we've had some luck when we don't have, like, we use a different gene, but like we have a similar 3-gene approach after you look at like the entire genome. Sometimes it can be helpful to look at the first 500 base pairs of the 16S. I know it's like seems completely useless for mycobacterium, but you get a larger database, right? Just because clinical labs have been sequencing that for so long. And sometimes that can be helpful, but the reality is it's just hard. And we have a tendency to report things out as most closely resembles because we don't get a definitive answer using any of the approaches. So it's curious. I wonder if adding in the tlyC gene would allow us to discriminate a little bit better.
Yeah, it's worth they had a poster on it at ASM a couple years ago. If you email me, I can send that to you too. But yeah, we actually, I did look at 16S for this once because coming from like a microbial ecology background, I'm like, well, obviously it's like a 16S. And it does, like you're saying, it actually does work pretty well. I've been told to kind of stick to these targets because that's the historical usage that our group has used, kind of getting out of my area here. But yeah, that's a good point.
And then I'm just going to ask a follow-up question. Do you guys have any plans? So I'm assuming you're talking about all these mixed strains. So you're going directly from primary specimen then, and not from clinical isolates that are growing back out?
Yeah, again, that's not 100% my area of culturing these mycobacterium, but I think, yes, oftentimes it's sort of a sediment, sequencing off sediment to try to get an answer as fast as possible, and then doing culturing, following up with culturing, I think happens a lot of the time. I don't really want to say that for sure. I don't know exactly, but that's kind of what I've overheard.
Okay, that's fair. Yeah, because you could test, I would think, at 3 steps, either primary specimen like sputum specimens that you're getting in, or once you have original growth out of the midget tube, which could potentially still be mixed, or you can test, like after you've actually grown midget back out on plates. So yeah, just curious, like where you guys are testing.
Yeah, I can't say for sure. Sorry.
That's fair. Thank you.
All right. Well, I guess it's getting near the top of the hour. Thank you everyone so much. It's been great.
Thank you so much. That was a really awesome talk. Have a nice day, everybody.